I decided to talk a little bit about the things, some of them that we've done and others in the room even, but to try to just sort of frame it at a very high-level not abstract, but functionally and anatomically. So the fine detail that you heard will not be present, so we just have to think about it. But let's see if I can, yeah. I want to just go back to the beginning a little bit of this field, what we call neuromodulation, which began really with very simple techniques of ablation or lesioning to basically address enormous problems that were confronting physicians, particularly surgeons who dealt with patients with extremely violent involuntary movement disorders, particularly tremor, which was undoubtedly one of the major concerns. This modern era that we've entered, I think, is really fascinating, and it's allowed us to begin thinking about modulation and very specific kinds of modulation of networks and dysfunction, and to, in a sense, restore function through some of these approaches. But a lot of these techniques based on stimulation have really evolved from that early history with ablation, and I think there are a lot of lessons there that we cannot forget and should not forget. And this was all early in the 50s and 60s, and many of the lessons were empiric, often serendipitous and quite interesting, how people stumbled into fields, but really striking to me how many of the targets that we use today were really understood and derived from that early work. Just to take a giant step back, this shows some of the targets of early approaches to, I thought I did it right. I think I just turned off my talk. Oops. There we go. Okay. Yeah, right. So early surgeons confronted with patients with extremely violent movement disorders resorted first to simple ablation of motor pathways, motor cortex, descending cortical spinal pathways. Of course, this was, well, partially successful in reducing tremor, had an enormous side effect of causing impairment of motility and coordination. And it was really the attack on the basal ganglia that opened the door to modern targeting. And this was done by courageous surgeons, I won't go into the detail, who really took upon a target that was considered untouchable because of the violent consequences, extreme consequences that could occur. But freehand, not in a stereotactic frame, but freehand, they targeted structures and identified the globus pallidus as a target. These are all basal ganglia, and the thalamus as a target, both for tremor and other movement disorders. In fact, pallidotomy and thalamotomy were done widely in the 50s and 60s. There were no other effective drugs available. The only drug was an anticholinergic, or anticholinergics that were used. But the discovery of levodopa in the brain, of course, and the successful treatment with precursor of dopamine and precursor of dopamine was transformative. I mean, this was probably the major discovery of all time in neurology, I would think, for treating neurologic disorders. It set up a model that has been hard to replicate for the most part. But this led to the abandonment, of course, of functional surgery almost all over the world, except for a small number of places that remained to treat tremor. And of course, then it became clear that this miracle drug, while producing dramatic benefit for many, many years, was then often complicated by the development of involuntary movements, dyskinesias, and then so-called motor fluctuations, the wearing off, the shortening of the response to medication. One of the questions I think underpins a lot of this, always to me, in talking with patients, is how is it possible that we can go to a very tiny specific point in the brain and either ablate it or stimulate it, and we get very much the same response. It's a remarkable finding, and it helps patients, I think, to understand that this is not all new, it's just different. We're doing it a different way. So I try to build on that in those conversations. And I think the answer is obviously what you would all know and say. It's the presence of very specific, well-organized, differentiated, functionally segregated neuronal pathways, networks, and particularly those involving the basal ganglia. So the basal ganglia, I don't have to tell too many of you, I think, what this means. These are structures deep in the brain, once characterized by their darkness. Kinnear Wilson likened them to a basement because of their darkness and untold things occupying them. But we now know that these basal ganglia are not really separate entities or structures. They are components of neural pathways that take origin in a number of places. One is the cortex, which sends topographic connections. The entire cortical ribbon sends anatomically organized projections to the striatum, the caudate, and the putamen shown here. And this is the important one for motor function because it receives the cortical spinal, or inputs from the sensory motor areas. So that's an input stage, and then the other input stage is the subthalamic nucleus, which has gone from bit player to central stage in our thinking about basal ganglia function, and it's both a major input area but also an output area. But the real output comes from the internal pallidum, which sends its output both to the brainstem and to the thalamus. These are all look like they're all scattered about haphazardly there, but I'll show you that the output from the cortex, most of it, importantly, from the frontal lobe, takes place from these different functionally differentiated cortical sites. I've just very schematically here. This is a monkey brain, a little easier to visualize. Premotor areas, eye fields, frontal and supplementary eye fields, prefrontal areas involved in executive function. This is the higher functions of the frontal lobe, all that we do and think, how we think and behave and structure our behavior. And then these areas that we call limbic, which are involved, of course, with mood, reinforcement, reward, and those types of behaviors, addiction as well. So that's I like to think of both the input areas from the basal ganglia, because basal ganglia, although they receive input from all cortical areas, they project only out back to the frontal lobe. And one of the things that we found when we began studying this was what the basal ganglia do, particularly what do they contribute to movement, was to the shocking sort of discovery that these sensory motor areas in the cortex were maintained actually throughout this entire circuitry, through the putamen, the pallidal segments, and the subthalamic nucleus, and that the output to the thalamus was also maintained. And I say that maintained in the sense of functional specificity, very clear relationships to direction of movement, individual body part, and a somatotopic organization. So it wasn't that there was a funneling of information, but rather a maintained segregation and operations obviously being performed on that network. So this was a motor network we discovered, but it wasn't the basal ganglia, it was just a part of the basal ganglia, a specific region of the basal ganglia, and that we came to realize this a period of time, was organized, the basal ganglia were organized as components with multiple parallel pathways originating in those different cortical areas, very separate, this motor circuit, very separate from the sensory and motor area. We now believe that we can think of disturbances arising in the motor loop, if you will, the motor circuit, as resulting in most of the disorders of movement that we think of, Parkinson's, chorea, dystonia, and others, bilismus, dyskinesias result from disturbances within this network, whereas the psychiatric behavioral disturbances that are associated with basal ganglia, obsessive compulsive disorder, addiction, and mood represent disturbances within this network. And these are really shown here, very closed loop, as you can see. The question of whether there is some overlap or integration remains always a question. But I think basically this model has held with imaging and other techniques. So I think it's got legs, as we say in the South. Still alive. So there's a framework to understand how, in a disorder like Parkinson's or Wilson's disease or Huntington's, with basal ganglia disturbances, you can see any kinds of admixture of these different motor, executive, cognitive, behavioral. And then we can think of these as network disorders, circuit disorders, if you will, each with a very signature characteristic type of activity. And we've studied all of these, dystonia, Parkinson's, bilismus, and they're all different. So the network activity is different. I'm not going to go into great detail. But obviously this provides a setup and understanding of how functional innervation that's very localized can interrupt and or modulate specific parts of the pathways. Parkinson's is just wanted to show you a little bit of an example of what we call Parkinsonism, are the cardinal features of that disorder: slowness, movement, difficult initiation, tremor, rigidity. And then there are non-motor features that are not dopamine responsive, so some of the gait and balance disorders, freezing of gait in particular, but a whole host of other things below the tip of the iceberg here of cognitive, behavioral, autonomic aspects, which we'll not really talk about, but just to mention. So when you look in the brains of animals with Parkinsonism produced by toxins or patients, indeed, they're pretty much the same thing. What you find in taking the three key structures, I think, in this, in terms of the output from the basal ganglia, you can see that the normal pattern here and a representative abnormal one here is increased firing. And a lot of variability and patterning that's not so evident here, but back in the subthalamic nucleus, you see the increased firing, but also the coupling, synchrony, which is quite abnormal, and the bursting, the oscillatory sort of patterns. There are similar changes in the external palatum, but the direction there is to decreased activity. So in the basal ganglia in Parkinson's, as we see it, there is increased output and there's abnormal patterning and synchrony that seems to be the hallmarks of this disturbance. And much of this work was done in the context of circuit models that were being elaborated around this time. This is all in the turn of or about 1990s, when this kind of models, Albin, Young, our group, and Crossman, a whole bunch of people, came up with kind of thinking that what has evolved now is that a model of Parkinson's where the subthalamic nucleus is excessively active and driving by glutamatergic pathways the output from the internal palatum, which is an inhibitory. This is a GABAergic pathway, so glutamatergic excitatory drive, and then excessive inhibition on thalamus and also on the brainstem in this interesting PPN area. So the model dopamine, I didn't comment on, but dopamine deficiency here, shown by the dotted lines, leads to this excessive activity over this arm of the pathway and decrease there and excessive output. So it makes it understandable in a way that interruption of the output in the pallidotomies could be very effective. I mean, it all kind of provided a basis. Of course, what changed the whole era really was the things that happened around that time. with the development of high-frequency stimulation. The work of Alim is going to talk us through that. Dr. Benabid, first in the thalamus for tremor, showing that high-frequency stimulation was as effective, equally effective to ablation, and certainly safer and more easily adjustable and reversible. And then we had the development of the primate lesion, or the primate lesioning of the subthalamic nucleus, which offered a new opportunity, but felt to be much too risky. And so we kind of went into a return to the Back to the Future with a return to pallidotomy, but much more carefully guided and functionally targeted to the appropriate spots. And so Laurie Leighton was one who had maintained that going. So a number of factors all played together here. And then, of course, GPI pallidotomy we did shortly, and then with the successful application to stimulating the subthalamic nucleus, this was the trigger for this renaissance in functional approaches for all kinds of movement disorders, dystonia, tremor, and now explored for epilepsy, and of course, psychiatric disorders. I won't have time to talk too much about that. So just to say, one of the things that are obvious, what's called the paradox of functional surgery. How is it that we can, with a small lesion or stimulator, reverse these monstrous signs and symptoms that ruin people's lives without incurring a debt, any kind of price to pay? Is it true? And then how can it be? This is kind of a topic that has long existed in neurology, because it's so obvious. That's just the GPI, lesioning pallidotomy approach, the question. And so we now have, just to summarize, a number of targets that we can use. The subthalamic nucleus for all of these disorders. The internal pallidum, pretty much the same story. There are debates which one is better. Both are used. The output is used for some, sort of broken this down, because there's a cerebellar recipient port, which is more the one used for tremor than of all kinds, but this one can be used. And then there's the downstream projections to the PPN, which are really fascinating, and this appears to be one of those regions that the basal ganglia talk to and are really very integrated with, not shown here in any detail, but that are important for gait disorders, freezing. I'm going to show you just a little bit of an example of the kind of things that this can do. When we stimulate something like the subthalamic nucleus there, and as you know, these are implantable devices, very much like cardiac pacemakers that are externally programmable. You can set here the frequency, the amplitude, the pulse width. Most of these parameters, without spending time, favor axons, the long connections rather than cell bodies. But obviously, this is a reversible, adjustable, far less invasive than lesioning, but has cons of having hardware, programming visits, access, availability. Have to have that, and battery replacements. But let me show you. There you see, that's the business end of this electrode as we currently configured, and this is a device that gets planted like a cardiac pacemaker, externally programmed. So I want to just show you an example of this. Yeah, so this patient, you don't need the sound, has severe tremor of the type I imagine surgeons may have seen decades ago, or hundreds of years ago. His left stimulator was just turned on, and now his right will be turned on. You won't hear this conversation as often, that's fine. But you will see how this dramatically disappears with stimulation. And so we run this continuously, turn a switch, and there it goes, and that's it. So there's no further adjustments needed. These can all be changed. It could be bipolar or monopolar, you can change it, but it just goes forward. One other example where you see better the effect. This patient has, and these are patients, both are off medication, so this is just stimulation. This patient can't get started. He's freezing. His gait is halted. He was given a cane there, and that helps, an external kind of stimulus. So now you see him after surgery, and this is a very short clip, just to give you the sense of the dramatic turnaround that occurs with these. So, come back a little bit to the paradox that we can do these interventions that restore function. They eliminate signs and symptoms, but they also restore function. Okay, I'm almost there. But I think you will agree, these must give a very exaggerated picture of what the basal ganglia do, since when you interrupt that outflow from the basal ganglia, either by lesioning or stimulation, there's not much to show. It appears to me, I think most of us, that what we're seeing with these signs and symptoms, the tremor, the difficulty initiating, executing movement, these are not the functions of the basal ganglia, but the disruption of function present in downstream networks in the brainstem and the cortex. So how does this all work? I can save this for questions, I think. There's some evidence that it might act like a functional lesion to sort of block transmission. A lot of ways we can think of that. It might be overriding abnormal activity, capturing it at high frequency. It has to be at high frequency to just cancel out or override. Activate, it does activate antidromically, which is another issue, and reducing phase amplitude coupling. We won't talk about that for sure right now. So it seems to me that both of these procedures, ablation and stimulation, are really not disease specific, but circuit specific or network specific or sign specific. And they're really the same targets that we use for Parkinson's, dystonia, and Chorea. They're all the same. We use the same. Part of it is a crudeness of, I'm going to skip that. I'm going to just add up, say a word just about potential future things. I think a lot of technical improvements are really underway, and even closed loop stimulation now is being tested, so we don't just turn it on, but we have either adaptive, so the best would be adaptive, that responds to the magnitude and severity and presence of that. And also better ways of stimulating with novel stimulation paradigms. These are the two areas, I think, that are really quite exciting, not just continuous stimulation, but pattern stimulation timing. But I think there's a possibility that we may actually go back to the future to some extent to ablation, which proved itself to be quite successful. It had side effects, of course, but now this can be done in a way that is very much less interventional, using focused ultrasound. And these studies are now underway for tremor, and they could be adapted to other targets as well. So I think that's potentially our future, short term. Right.
Okay, we have time for a couple of questions. Could you just raise your hand again so that the mic will know to come to you? And please identify yourself too, okay?
Okay, my question is whether or not, and you raised this, whether or not both ablation and high-frequency stimulation disrupt the function of the basal ganglia. So can you not image the places where it normally feeds forward to and find out whether that is indeed the case, that both disrupting to the same extent?
Yeah. You know, the very interesting thing is that when you do image, and we've done the imaging pre and post, what you do is normalize, in the broadest sense of the word, the cortical areas that are disrupted, and you remove well, not remove, but the areas that may emerge and appear to be either compensatory or even dysfunctional, but you remove those areas. So this procedure, ironically, has a big picture of reversing the abnormalities that were present in the cortex. But the bigger question you ask, I think, is what do the basal ganglia do? And what have we really lost? I think it is a huge question. Some of it has been answered away in the songbird that Corey mentioned. You can lesion the basal ganglia in the songbird and produce virtually no effect on learned behavior, learned song. They learn the song. It may degrade somewhat over time, but if you lesion it before they've learned the song, they have trouble learning. And my firm belief is that the basal ganglia are primarily involved in learning, both motor and more complex forms of learning. I think they're also very important in motivation and sort of get up and go. But this is a very complex area. I think they are less involved with movement in any obvious direct sense than we thought. I think that they are not involved in really ongoing motor activity. Are they not a storage site for movements that were learned or innate? They are involved much more in the dynamic process of responding and moving, or learning, and setting up for future responses. This is another whole lecture.
Tom Klein, I was wondering, my impression is these treatments do wear off with time. Is that correct? And does the deep brain stimulation wear off, or does the ablation wear off?
Well, I think it depends where you're stimulating and what you're stimulating for. There is certainly, for thalamic stimulation patients with central tremor, so-called, and others, there can be waning of response and changing of response characteristics. And I think that's striking to some extent. For dystonia, the responses are really quite stable once they're established.
The ones that are unstable, I'm wondering if you follow the deep brain stimulation, once it wears off, will then ablation restore the
Oh. Well, you know, I think there's one problem with stimulation is that you may not be optimally on target. There are three things, or several things that influence. One is selection of patients, you know, the right patients. You've got to get those, and then you've got to put that electrode in the right spot, and then you have to program it correctly. I mean, this is an art and a science of programming, and I think some of the wearing off that patients see is really just poor placement or poor programming. It's a complicated issue, often to sort out.
Sort of relevant to that last question, to what extent can you make the programming process something the patient participates in, as opposed to something that happens in the doctor's office once, a month for 10 minutes?
Yeah, that's a really important point. I think it's where things are going. Patients who now have the ability you can give them options so they can increase their amplitude. You can change frequencies, those are the two probably main ones, I think, and patients can then vary. And there are a few patients who do better for speech with one setting and for walking with another setting, so they can switch between. This is not ideal, obviously, but in some cases. There probably is also, I think, an argument for more than one electrode placement, that we can probably get more by, say, targeting two different parts of the single structure or two structures. STN or the centromedian is another, getting down deep in the weeds, but yeah.
Okay, thank you very much.
Thank you.